way we communities trying to understand that is we look at the structure, right? You know, the computer, we look at the wiring of the components as well as its function. And on the structural side, we actually love putting on Switzerland over the last decade, made a lot of inroad into understanding small organisms. We have the wiring diagram of all hundreds of neurons in the worm, or live, and so on. So that's all been done with electron microscopy, which you're going to see actually as a next stop. The challenge is that these electrons, they don't go in inside the brain, they don't go through. So what you need to do to study is actually cut it into thin.
You're actually going to handle the spike.
Very small slices. Yes, 40 nanometers, millions, that's the millions of things. In order to go through a single millimeter, you need tens of thousands. It's very difficult to study large groups. The human brain doesn't do it. So all this, the technology is developing such that we get slowly up to the resolution we actually need to see what is going on, see how all these cells are wired up, see all these fibers that connect between individual neurons. We hope that these approaches will allow us to understand how brains are wired up, how they're differently wired up in different disease conditions as well, and maybe also learn about natural intelligence and how it actually comes about from wiring. But that's one side. The other is really the function of the brain, the electrical activity of all those neurons. And we and other groups develop kinds of technology to put in wires into the brain, some to just do our research in the lab, but also, one startup coming out of my lab is now aiming to put a device developed into patients that have a brainstem strokes who can't speak anymore, but can form those words and record activity in those areas to allow them to speak again. Hopefully in a year we will have the first of those patients. And my lab itself, I have 2 neurosurgeons as well. It's one of the benefits of the Crick, really working very closely with the clinic. And those surgeons are now putting tiny wires into patients that agree to these trials while they are. We hope to understand how their brain activity is maybe different if they suffer from certain diseases compared to others. And sort of overall of these beautiful pre-found, I think it's right to...
Presumably if we can understand more of the wiring, we could also, obviously the disease part is speech itself, but in terms of our emotional regulation and that stuff, because that helps understand mental health and what stuff too.
I'm a very discovery scientist. Yeah. We have to really understand it to treat that. I initially are a bit closer to that, and I think that's exactly what we are trying to do.
It wasn't a deliberate segment.
No, but you're brilliant.
I'm trying to think of some good questions to ask.
Yeah, because I'm actually a psychiatrist. Psychosis. So just to give you a brief introduction into psychosis, imagine a young man, let's call him Jake, in his 20s. Jake is about to graduate from university, is thinking about proposing to his girlfriend, so life is looking good for him. But then something shifts. And it's sleep, and psychosis is all the and what it's often like, often when life is unfolding, it's more often than not definitely. Hello, very nice. How are you?
Nice to see you.
And you. So welcome to Electron Microscopy Science Technology Platform. So I'm deputy lead here. And what we do is we work in routine projects. We have quite complex projects, and projects last between 6 months up to several years. And they're all pretty much bespoke. We have lots of different types of technology, and they work very closely with the group leaders. So the sort of samples that we use, we look at cells, so basic unit of life. So it could be individual cells, or cells obviously sort of forming tissue. Which is you get, in your biology textbooks, you get pictures of a cell. That was obviously happening.
That's my biology A level.
Nucleus, mitochondria.
Yes. Yep.
Good. That's the sort of thing you get. And what we're going to do in a second, we'll go to do in a second. But in order to get the samples into the microscopes, the inside of a microscope is very inhospitable. It's high vacuum. We bombard it with high energy electrons. So we have to do an awful lot of sample preparation. Because we don't want our nice little soft biological material going into a vacuum. It doesn't tend to matter. So we have a whole huge process with chemicals, etc., but for samples that go into the TEM, which we're going to look at in a second, we have to then cut very, very fine sections, which is Dumi. She is going to tell you. So this is Dumi Nukana.
Hello.
She is
That was funded by the CCI. So that was then to build a pipeline
where we democratize access to. So the point is that they can be used in low-cost settings or in remote facilities where people cannot get to put expensive machinery or change the environment to hold them. Used there for local research. So because I don't have much time to get more into details on that, the fun part begins. So I'm going to need your help.
Sure, yeah.
So
Make sure your fingers don't get cut off.
What's actually in there? What are we looking at?
So this is my block, and we're ready to section it. It can be anything. It can be brain tissue. Upstairs you already heard about the connectomics project. So all of that can be cut and then basically put together again after you image to create a volume. So it's kind of like working backwards. But the point is that you can then put all of these together to create a volume. Most important is that you pick them up in a serial format. So you need to be able to look under the microscope and know which one came first. So then you can pick them from 1 to 1,000 or whatever ridiculous number, 100,000, because depending on how much of the tissue you have. So these ones are all over the show. So if we press here, so I've set the start and the stop for the microtome basically to kind of like slow down as it hits the knife, because you don't want to damage that and you want to cut it nicely. So sometimes it takes a while to basically produce a section, but we already have ones cut for you. So what I will need you to do is take a sip, and then you can adjust then the eyepiece so that you can be able to see and keeps cutting. But if you stop it, it takes a while to again start like cutting.
Okay.
So it's either if we can wait for that advance. If we run out of patience, we can move along.
This is 40 nanometers.
So these you can cut down to 30 nanometers.
30.
Yes. So if you think of a cell, which is like probably can go to 10 microns, and then you have to cut it at 30 nanometers. So you're going to end up having like 330 sections that you need to pick them up. If you're cutting the tissue, then it's a whole lot. Some people do a whole worm, which is going to be like thousands of sections that you put up. I think there you go now. It's about to cut. Yes. Until it is up. So now that we have quite a few, this is your eyelash.
Okay.
Now, what I want you to do is moving the sections.
There's a hair on the end, is that right?
There is a hair.
It's meant to.
Eyelash. It's meant to be.
Oh, it's
hair.
meant to be. That's it.
Yeah.
When you said eyelash, it literally is that.
It is an eyelash.
Okay.
Yes. We are very obsessed with eyelashes. Everyone with long eyelashes will like, can we have some? Yeah. So we're going to stop now, and then you can move them. The only thing that is important is that you don't poke the sections because they can tear up.
Okay.
And also sometimes they curl around the eyelash. So what you want to do is either you maneuver them at the corner or you use the water to roll it back so that
You didn't tell me this is like a really serious research
Yes.
project you're doing.
We have a replicate. Don't worry.
I'm not responsible for messing this one up.
Yeah. So here we have an ITO, which is
Okay, so I'm brushing them back.
You're brushing them back
Okay.
in a sequence because I want
You
them
want them all together?
all to the back.
Oh my. All together, they can't break up.
You can break them up as long as you can put them together again in a sequence.
Well, I'm moving on in a sec. Okay, let me see.
She's a hard taskmaster, isn't she?
It looks
wow,
so
yes.
weird in the. Okay. Is it best to have it facing upwards or downwards?
Gosh,
Whichever way
this-
The ones that do.
Okay, so these are the students from William Ellis School. My colleague Ellie here is going to introduce you to the ones that are going to be chatting with you today.
Super.
Fab, do you want to take
Hello,
a seat?
seat.
We've got you some gloves,
Thank
we've got
you very
everything ready.
Excellent, ready to go.
And I can introduce you to, we've got
Nice to meet you guys. What are you learning today? What's going on?
PCR.
PCR. Is that something to do with swabs and? COVID, I remember PCR from COVID days.
Yeah.
Is that?
Yeah, basically that.
Is it the neutral sample? Is what it's called?
Neutralizing?
Is it neutralizing? Is that one?
It's not that one,
Okay.
but
What's the PCR then?
polymerase chain reaction.
Okay. Can you tell me what it?
You've just modelled it over there.
Yeah.
So what are you doing in the model?
So you're breaking up DNA chain and then primers attach to the DNA and like it's all so that we can use this in the production of insulin through E. coli. And originally it was produced from pigs and how we remove the bacteria off due to ethical reasons or something.
Amazing. And so what do you have to do? Do you have to actually draw it out of a substance, or is it to go into the little pipettes?
Yeah.
I think this is where you get your chance.
This is where I have it as well. Okay. You should.
So you take a micropipette.
Yep. Goggles, his glasses. I'll get the glove on first, then put the glasses on.
And sorry, just had to remind you. I'm sorry.
Glasses on.
So you take a micropipette.
Yep.
There's 2 stops on it when you press down. You can hold this one.
Yep.
Yeah. That's the amount. You take it off when you stop pressing down afterwards, and that draws in the liquid. And then when you're ready to put it into the next like the cylinder, you press down to the second stop, and that dispenses it.
Okay.
And then the white tab here, you do that to get rid of the tip. You press that to get rid of the tip.
Gotcha. That's it. That's it. So that goes in there.
Yeah.
Gotcha. Press the.
So don't press anything.
Just like that. Okay.
It's also best to hold it.
Before you go and hold it to the plastic.
Push the. Here we go. And then release.
Yes.
Okay.
And then take it out.
Ah, we've got something good.
And then place it on
here.
Yeah.
Pressing back in again.
And
Press it all the way through.
All the way through. Yeah.
There we go.
And then use that white to get some.
Like that.
Yeah.
Hey. Okay. Thank you.
Perfect.
So what is that I put under? That's dye, is it?
This is just
Food dye.
Okay.
That was just a practice earlier with some food.
Oh, okay. Yeah, I would not trust anything too special today. It's good news. I was looking at the electron microscope downstairs. That's like quite a big bit of kit, quite a serious bit of kit. Don't get me touching that. Getting expensive. Are you enjoying learning though, guys? Are you all into science and kind of research?
Yeah.
Okay. What are you all studying?
We're all A-level biology students at the moment.
Where are you all at the same? Where are you at?
We're at the swap.
Okay. Good. So actually coming here and doing the practical work is quite exciting, quite interesting.
Yeah.
Do you get to do much practical work where you are?
Yeah.
You do. But here's different because
Well, we get
you get more stuff to do, more fancy equipment. You get Charlotte helping out.
Yeah.
Good. And Charlotte, what sort of things do you try and expose them all to that they may not get a chance to do elsewhere?
I guess these kind of pipettes here are quite interesting. There's different amounts.
What's a vortex mixer?
Don't forget to like this video and subscribe for more royal insights. Until next time, stay regal and stay curious.